Welcome to the add-on course of instrumentation course in life sciences, which is given by Vivekanand College, Autonomous Kolhapur, Department of Biotechnology. I am Vinaya More. I am taking the syllabus from Unit 3, which consists of instruments used in microbiology lab. Microbiology is the branch of science deals with study of microorganisms. Microorganisms are those organisms which are does not seen with naked eyes, means they are seen with only with the help of microscopes. Microorganisms are too small when they are grown on a solid media, they develop a colonies and these colonies are counted, means these colonies or number of colonies are measured with the help of one instrument, that is colony counter. Today we will discuss about types of colony counters, on which principles colony counters works, parts of colony counters and uses of colony counter. A colony counter, its name suggests that it is the instrument or device that is frequently used to count bacterial or other microorganism colonies which are developed on a plate that contains a gelled growth medium. But one microorganism cannot give rise to one colony after completion of its cell cycle, but one colony it is produced by multiple cell divisions of one microorganism, or one colony is the population coming from a repetitive cell division of one bacterial cell or one microorganism. A colony forming unit or CFU is a precise measurement to determine how many bacterial or mycelial cells present in a given sample. Thus, the colony counting approach monitors the viable colony forming units which can still reproduce via a process like binary fission under a careful monitorized conditions or settings. But next is purpose of colony counting, or why colony counting is important. To determine the approximate number of cells in a different samples, colony counting is used, or when we can culture a microorganism at a specific temperature or at a different quantities of nutritional medium, then how many number of cells can be grown, which can be determined with the help of colony counter. Ideally, a single viable cell can undergo massive replication to develop into fully developed colonies in the form of CFU, or these fully developed colonies are measured in the form of colony forming unit with the help of colony counter. Next, but there are some issues with the colony counting, or there are some cares which to be taken during the colony counting. First, the number of colonies that do not overlap means during counting one colony can be not repeatedly counted, means number of colonies that do not overlap. The number of colonies that contact with edges are also counted or also measured during the counting. It to reduce the count because of noise. During the colony counting, the noise to be created which can give rise to false positive result. So care to be taken that to reduce that noise, so accurate results are obtained. Colonies of various size, shape and colors are also produced during growth conditions that to be counted during the colony counting. There are some importance of colony counting is that this in different businesses like food industry or beverage industry, the number of cells which can produce a product are measured with the help of colony counting. Determining the colony counts helps to make ensure that discharge of product wouldn't eventually cause any harm. Means if we can calculate how many number of cells are present in that media, then how many cells produce a product can be concluded, or amount of product produced during that conditions can be concluded. During once we can determine the number of colonies in the blood or any samples which can use the results in a medical microbiology or in a pathological laboratory, that is how many number of cells or how many number of bacterial cells are present in the patient's blood sample for giving the results, that is how many number of bacterial cells are present in patient samples. Next is there are different types of colony counters. First one is manual colony counter and second one is automatic colony counter. On a screen, both the images of colony counters are displayed. First one is manual colony counter. In this colony counter, the plate on which the microorganism can be grown, which is placed in a plate holder, and with the help of pen on each colony, the pen is tapped, and during tapping the noise is created which is measured by that machine, and colony number of colonies, that is colony count to be produced. But in the automatic colony counter, as we can see on the screen, the photograph of plate can be taken, and from that photograph by using the software number of colonies are counted. During the colony counting, in manual also and in automatic also, triphenyl tetrazoium chloride, that is TTC, was incorporated to make the colonies more apparent, and because live bacterial cell convert TTC to deep red colored product, so that colonies, bacterial colonies are more bright as compared to the background, which is makes easy for colony counting. First is the manual colony counter. Manual or semi-automatic counter rely on the technician's ability to distinguish between colonies and mark them on plates outside using a specific pen. The device maintains a track of colonies with such marking, although this approach somewhat aids, means it is time consuming and prone to provide a mistakes. But there are some specific parts and their functions of manual counters are as follows. First one is auto marker probe pen, which is used to count the colonies. That is, a counter pen's pressures on a colony causes a count to be registered by a audible beep or sound and advance on a computer screen. Digital display, it reflects the total count made by the counter pane, means noise created by counter pane. Lenses which carry out magnification and light source provide a light for illumination purpose. Working principle of manual colony counter rely on a Petri plate is placed on electronic pressure pad with a light illumination, and each colony on a plate is marked by tapping the plate with an auto marker probe pen. A count is registered in the digital display by the touch pressure. This pressure can be altered based on the situation. It facilitates the avoiding the error of overlooking or overlapping of the counts, means counting colonies twice. The equipment also a gradual for a segmentation disc and centering adapters for 50 to 90 pads. Additionally added features include darkening background for transparent colonies, free illumination that allows a optimal peripheral colony viewing, and an integral over averaging tool for multiple plate counting. But there are some limitations while using manual colony counter is that it is intensive, means it requires labor or technician to count the colonies. It is time consuming, gives relative low through output and high risk of human error. So to overcome these limitations, scientists developed automatic colony counters to effectively count the colonies. Autonomous bacterial colony counter employ a image processing method. Means this automatic colony counter based on the principle of image processing, which consists of following part. First one is culture dish. In a culture dish, culture to be maintained or bacterial samples to be cultured in a culture dish with the help of plate inoculation method or spiral inoculation method. Light source, a durable LED light source is used, which provides a 4 different light sources and background color combinations for adjustment of light. Optional intelligent remote control multicolor light source is also provided for improving the background accuracy of the count of various media color and the type of combinations. Third one is imaging. The conventional artificial colony counter uses 3 to 6 times magnification lenses for imaging. And last one is image processing. The automated colony counter has a picture processing feature. It carry out background processing, color making, interference correction, colony expansion, area calculation and other elements which are not seen in conventional colony counters, which are provided in the associated software. Automated colony counter improves the data source, intelligent query and data expert with the help of software. Some can also operate permissions, data modification permission, etc. to make the measured data accurate. Additionally, you may print and edit the reports online simultaneously. Means there are many facilities are provided in automatic colony counter as compared to manual colony counter. Next and very important is principle of automatic colony counter. How it works or how it can carry out the measurement of colonies which are present in the Petri plate or which are present in a culture. A computer-based system is fundamentally composed of collection of image utilizing any of the digital image capturing device such as document scanners, charge coupled device that is CCD, digital cameras, webcams or video equipment. Colonies can be enumerated from an image of plates through the use of software tools. Means in a simple language, first automated colony counters take the image of plate on which colonies to be counted. Every plate is photographed and then analyzed. The recorded photos are then converted into digital files on a computer using a programmable image processing software. A single or multi-threshold segmentation techniques are used to separate and identify the colonies which are present on the plate. The background of object and object themselves can vary greatly in automated system. To improve the visibility and boost permission, one of the three illumination techniques can be chosen. Means automated colony counter provide 3 different illumination techniques, means light sources. First one is transmission technique, second one is reflection technique and third one is dark field technique. In transmission technique, which is applied common, high contrast atoms with largely clear backgrounds are used. Second, reflection techniques which are used for object with high contrast and opaque backgrounds. And third one is dark field technique for low contrast and largely transparent objects. There are some advantages of automatic colony counter over the manual colony counter. First one, it can use accurate result from most difficult samples. It work with any samples in very little contrast. It take on colonies of every size and form. Take care of all noises. Even in extremely overlapping colonies, high curvature locations can be taken. Click a photograph of petri dish containing a colonies in a 10 second and software can evaluate immediately, means it reduces the time required for counting. The time needed for counting is reduced and the use of software decreases the manual effort, means it does not require any technician or label for colony counting. There are some applications of colony counter. First, it is more useful in microbiology lab to count the colonies which are present on filter or membrane, which can determine the capacity of retention. It is crucial for determine the efficiency of disinfectant, means it is used in sterility testing or safety of food and drug, in medical examination assessment or in pharmaceutical industry. Measure the density of microorganism in a agar plate, slide mini gel or in a petri dish. It is used in AIMS testing. AIMS test is carried out to determine the carcinogenicity of any chemical. And last, it is used in bacterial mutation assays. This is all about colony counter, which types of colony counters are used, how they are constructed, how they are worked, etc. Thank you.